So our presentation entitled What Do Genes, Voltages, Behavior, and the Hippocampus Have in Common is analyzing a paper looking at voltages as they relate to behavior changes within the hippocampus. And so this presentation gives an overview of the research article Voltage Imaging and Optogenetics Reveal Behavior-Dependent Changes in Hippocampal Dynamics by Yoav Adam et al., published in Nature in 2019. And so before we get into the paper, we have to understand what the hippocampus is. And the hippocampus is a structure in the brain associated with storage and production of memory, as well as some aspects of emotional processing and behavior. And if we take a deeper dive into the hippocampus, we can see that it's composed into distinct layers. And this paper analyzed the two outermost layers, the oriens in blue, SO, and a deeper pyramidal cell layer in red, SP. And the two different behavioral states that the paper looked at was the quiet and walking state. And quiet just represents when a mouse on a treadmill is not moving, when the treadmill is not operational. And then walking represents when the treadmill is operational and the mouse is walking. And so the first piece of information that the study presents is that in the pyramidal cell layer, in that deeper layer, as we go from quiet to walking, there's an overall less activation, or as we call it, inhibition of neurons. And this is just seen in the right image where we could see that as we go from quiet to walking, the amount of spikes is lower, signifying that there's a lower activation. But in the bottom, in the blue, in the oriens layer, we can see that the opposite holds true, where as we go from quiet to walking, the amount of spikes increase, signifying that we're getting more activation. And this is just seen in the right image where in two different layers, where we could see that there's an overall increase in the amount of spikes, signifying overall increase in the amount of activation of neurons. And this is just corroborated in this figure, where we can see in red, in the pyramidal cell layer, there's an overall decrease in the firing rate as we go from quiet to walking, and the inverse is true for the oriens, where there's an overall increase in firing rate as we move from quiet to walking.
We can use a tool called a genetically encoded voltage indicator, or GEVI for short, to measure the potential of a cell. A GEVI is a protein that changes its brightness based on its surrounding potential. The video you're watching right now is a GEVI in action. Notice how it is glowing and getting dark because the voltage surrounding the protein is changing. You might recognize that this picture depicts a voltmeter and it is used to measure voltage. In a way, you can think of a GEVI as a voltmeter for the brain. The picture on the left shows a voltmeter in action and the picture on the right shows a GEVI. The main similarity between them is that they measure potential. However, they measure it through different ways. As you may observe, a voltmeter is plugged at 2 points of the circuit, as pointed out with the red arrows. Meanwhile, a GEVI is injected into a cell, as highlighted with the blue arrow. The green arrow highlights the camera, which is used to measure the fluorescence. So the big question is, does measuring electricity through light really work? In this picture, the red curve measures the change in brightness of the cell, while the black curve measures the change in potential of the cell. There are two different measurements, but they look virtually identical. In this second picture, the curves are overlapped, and you can see both measurements follow precisely. They use another measurement to confirm that the change in brightness is related to the change in potential. The researchers used signal-to-noise ratio, or SNR for short, of the light and compared it to the change in potential. As you can see, when you increase the potential as indicated when you go to the right of the graph, the data point tends to be higher, which means signal-to-noise ratio is greater. In other words, the potential and the change in brightness are strongly related to each other.
So the neurons were viewed using a camera and two lasers. A pattern red laser at 640 nanometers and a pattern blue at 488. The red was used to excite the neurons of the brain, and without getting too much into it, the frequency of the red light allows cells to produce more energy, and thus there's more actual potential, which the GEVI can then track. The blue laser was simply used for illumination and was overlaid with the red in the same pattern. Now, this blue and red was used since our brains do not like interpreting the colors of red and blue next to each other without there being depth, which is the same idea used for 3D movies. In the graph, we can see that once the blue light was added, the signal-to-background ratio, which is almost exactly like the signal-to-noise ratio, that is useful information, we can see that it was increased by about a factor of 3 or or 5. Now here we have footage from an Xbox Kinect, another older technology. In this, we should see patterned lighting. This is, instead of shining one singular light over an object like a flashlight, it is shining an array of lights in a very specific way and then tracking the movement of each of those individual lights in order to get data. A more comical example would be something like this, toys. So we see many, many little pins, which allows for more detail, and something like that. When we put it all together, we get an image like this. The wide field is simply when you shine light from below the object and look down at it like in a microscope. We see with the red and blue that the image was clearer, but with the PCL, it was hazier since it is a deeper layer of the brain.
So why was this paper important? Well, this paper allowed for the presentation of a new powerful imaging tool that allowed for the long-term analysis of voltage using genes. And what we can use this for is this can provide insights into the elusive mechanisms behind learning and memory within the hippocampus. So we could learn how learning and memory actually occur over a long period of time.